Welcome to another video tutorial on morphology and physiology of bacteria. So let's get started. So first we'll study on morphology of bacteria. Okay, now what's morphology? So morphology is the study of the external features of bacteria. So what we see externally, like it may be size, shape, arrangement. Okay, all these comes under morphology. So starting with it, the major characteristic of bacteria are based on their size, shape and arrangement. So mainly we study here on size, shape and arrangement of bacteria. So starting with the size, the unit of measurement used in bacteriology is micron. So yes, the unit used to measure bacteria is micron or micrometer. Okay, it's actually a very small unit. So I think, as we all know, that 1 centimeter is made up of 10 millimeters and 1 millimeter is made up of 1000 micrometers. So you can just imagine that micrometers itself is a very small unit, and the unit of resolution with the unaided eye is about 200 microns. Okay, so that means our human naked eye can visualize only up to 200 microns. Okay, or I can tell it can distinguish between two closely spaced objects when it is 200 microns. Okay, when it is less than 200 microns, we cannot visualize through our naked eye, and hence to visualize bacteria we require a special instrument which is called as microscope. Bacteria ranges from 0.2 to 1.5 micrometer in diameter and about 3 to 5 micrometer in length. And yes, so what is the size of bacteria now? It is from 0.2 to 1.5 micrometer in diameter and 3 to 5 micrometer in length. So just imagine how small and tiny is bacteria. Okay, and that's the reason we cannot visualize them under our naked eye and we require a special instrument called microscope. So next is shape and arrangement of bacteria. So coming to bacterial shapes, so the first one is cocci. Okay, so cocci is spherical shape. So as you can see in the picture, if the bacteria has a round or spherical shape, we call them as cocci. And bacilli is rod shaped. See, a bacilli is something which is rod shaped. And next is spirilla, is spiral shaped, as you can see in the picture. So this is spirilla. Spirilla is spiral shaped. And next is vibrio. Vibrio is comma shaped. So you can see this picture here. So it just looks like a comma. So this is called vibrios. And then next is spirochete, flexible spiral shape. So yes, so spirochetes are more flexible when compared to spirilla, and these are also spiral in shape. And next is actinomycetes, branching filamentous bacteria. So this is actinomycetes. Okay, so they have branches around and they are branching filamentous bacteria. And then next is mycoplasmas, cell wall deficient and do not possess a stable morphology. So this is mycoplasmas and they do not have cell wall, and since they don't have cell wall, they do not have a stable morphology, that is they do not have a fixed shape, they keep on changing their shape. Okay, then so next is bacterial arrangements. Cocci can be arranged as following. So yes, that is cocci is round shaped or spherical shaped bacteria. So it can show the following arrangements. So first is coccus is single. That is only when there is one bacteria or cell you call it as coccus. And the next is diplococci. As the name tells here, diplo means two. Okay, and cocci is round shaped cell. So that means it is in pairs here. So as you can see in the picture, diplococci is 2, that is there is 2 cells. Okay, and then next is streptococci, that is strepto means chains. So when the round shaped cells are arranged in the form of chains, you call it as streptococci. So look at here, the cells are arranged one after the other to form a long chain like structure. So this is called as streptococci. And the next is staphylococci, which is grape like clusters. So this is how it looks. Okay, so the cells are arranged one after the other to form a grape like cluster. So this is called staphylococci. And then tetrads, as the name tells, tetra means 4. A group of 4 cells is called as tetrad. Okay, and then a group of 8 bacteria or cells is called as sarcinae. So this is the arrangement of cocci. And then coming to bacilli, when there is a single cell of bacteria, we call it as bacillus. So as you can see here, a single is called as bacillus. And then diplo, that is when there are 2 bacteria, you call it as diplobacillus. And then streptobacilli is when the bacteria is arranged in the form of chain, you call it as streptobacilli. And then cocobacilli, coco is nothing but coco here means spherical shaped, bacilli means rod shaped. So this cell will have both the features, it is mixed. Okay, so it is oval in shape and appears like bacillus. So as you can see in the picture here, it appears to be rod shaped and it has spherical shape also. So such type of cell is called as cocobacillus. And the next is palisades. So palisades are fence like arrangements. So as you can see here, they have arranged one next to the other to form a fence like arrangements, which is called as palisades. So next is physiology of bacteria. Okay, now what's physiology? So in the morphology, we studied about the external features of bacteria. So whereas in physiology, we'll mainly study about the internal functions and parts of bacteria. Okay, so which encompasses aspects like growth, metabolism, reproduction, adaptation, etc. Okay, so and in this video lecture, mainly we'll be studying about the functions, that is internal functions of bacteria with its parts. Okay, starting with that. So here, as we can see, this is the structure of a prokaryotic cell, that is a bacterial cell. This is how it looks. So it is very simple and it has some of the important parts like capsule, cell wall, cytoplasmic membrane, ribosomes, pili, flagella, nucleoid, cytoplasm. Okay, so now we'll be studying about each and every part in detail one by one. Okay, so starting with it, so first we will study about bacterial cell wall. So now where is it located? As you can see here, there are 3 main layers which is completely surrounding the bacterial cell. Okay, amongst that, one is cell wall. The green color thing which you can find here is the cell wall, and cell wall is one of the very important part of the bacterial cell. So hence we'll start with it. Bacterial cell wall. The cell wall is a tough and rigid structure surrounding the bacterium like a shell. So yes, it is very tough, it is very hard, and it is a rigid structure. So rigid means, so it is a structure which cannot be changed, which surrounds the bacterium like a shell. It is completely surrounding the bacterial cell. Okay, and it is protecting it like a shell. And then it is 10 to 25 nanometers in thickness. Okay, coming to it, so it can be about 10 to 25 nanometers in thickness. And then there are 2 different types of cell wall, gram positive and gram negative. So yes, so there are 2 different types. Not all bacteria will have a same type of cell wall. So it can be differentiated into 2, that is gram positive and gram negative. So we'll study on the differences between gram positive and gram negative in the following slide. Okay, and then the rigid part of the cell is peptidoglycan. That is the main component of cell wall is peptidoglycan. It is the one which is giving rigidity to the bacterial cell. And then what are the functions of bacterial cell wall? It gives shape for the cell. So yes, first of all, this is the one which is giving shape to the bacterial cell. Okay, it has one fixed shape and it is not taking up any shape it wants, is because there is a very rigid and tough cell wall present around the bacterial cell. Okay, when there is no cell wall present, no, so the cell can take up any shape it wants, just as we saw in mycoplasmas. So it can take up any shape because it doesn't have cell wall. Only when there is cell wall, because of its toughness and rigidness, so it gives a particular shape for the cell. Okay, and then it takes part in cell division. So yes, it's one of the important part which takes part in the cell division. When the cell is dividing, so this takes part, and then confers rigidity to the bacteria. So just as we spoke, it gives the rigidity to the bacteria, so it will not allow the bacteria to change the shape, and it gets fixed in one particular shape. Okay, and then it provides protection against osmotic pressure. Okay, and it also protects against osmotic pressure. Means osmotic pressure is nothing but the force applied to a solution that prevents solvent from moving across the membrane. It's nothing but the force applied by the solution. Okay, so that force of the solution also will be protected by the cell wall. Okay, then okay, now here we'll study the difference between gram positive and gram negative bacteria. Okay, so first is gram positive bacteria. So it has thick peptidoglycan layer in cell wall, and gram negative bacteria thin peptidoglycan layer in cell wall. So as I discussed in the previous slide, so peptidoglycan is the main component of cell wall, and it is the one which gives rigidity to the cell wall. Okay, and so in gram positive bacteria, no, so it has a thick peptidoglycan layer, as you can see in the picture over here. So this is your gram positive organism, and look at the peptidoglycan layer. Okay, it is this much, so it is very thick. Okay, it has a thick peptidoglycan layer. So whereas in gram negative organism, as you can see in the picture over here, it has a very thin peptidoglycan layer. Comparing with it, so this is very thin. Okay, that is the first difference. And then no lipopolysaccharide membrane, and in gram negative lipopolysaccharide membrane present. Okay, here in this gram positive you cannot find LPS layer, that is lipopolysaccharide layer, whereas in gram negative, see the here you can find the lipopolysaccharide layer, that is LPS layer. So this particular layer is not present in the gram positive. Okay, and then it produces exotoxins, that is gram positive bacteria produces exotoxins, and gram negative bacteria produces endotoxins. Okay, and exotoxins, as the name tells here, exo means these are the toxins produced outside the bacterial cell. Okay, and so they are released by the bacteria into the surrounding environments. So whereas endotoxins, as the name tells here, endo means so they are a part of bacterial cell structure. Okay, and they are released when the cell is damaged or destroyed. And then stained purple by gram staining, stained red or pink by gram staining. Yes, so when we do gram staining, that it is a type of coloring the bacteria. So when we do this staining, no, so gram positive bacteria appears purple. Okay, so whereas gram negative bacteria, it appears red or pink. And then gram positive bacteria contains teichoic acid. So as you can see here, so this is the teichoic acid, lipoteichoic acid. Okay, so this is the teichoic acid present in gram positive bacteria, and in gram negative bacteria there is no teichoic acid present. Okay, and then in gram positive bacteria outer membrane is absent, and in gram negative bacteria outer membrane is present. Okay, so yes, outer membrane, as you can see, is present in the gram negative bacteria. So see here, this is outer membrane, it's present in the gram negative bacteria, and it is absent in the gram positive bacteria. So these are the major differences of gram positive and gram negative bacteria. Other cell structures, cytoplasmic membrane. So cytoplasmic membrane is also called as plasma membrane. And as you can see here, so this is the membrane which is surrounding the cytoplasm of the cell. Okay, and so above this comes the cell wall and then comes the capsule. Now what is the function of this cytoplasmic membrane? So it acts as a semi-permeable membrane. Semi-permeable, we also call it as selectively permeable membrane, because it allows only the molecules or ions which is required. Okay, and it restricts the molecules and ions which is not required. Okay, it selects. So it selects which is important, which is required, and it restricts which is not required. Hence we call it as semi-permeable membrane. And then it also contains enzymes necessary for cell wall synthesis. So it also helps in the cell wall synthesis. Okay, and then next is cytoplasm. Now this part of the cell is called as cytoplasm. The innermost region you can see, the complete orange color thing. So this is called as cytoplasm. Okay, and then so the cytoplasm is actually a gel-like substance. So here completely a gel-like substance will be present. So inside which there will be organelles like ribosomes, mesosomes, vacuoles, and inclusions, and all those things will be present in the jelly-like material over here. Okay. So first is ribosomes. These are responsible for protein synthesis. So as you can see in the picture over here, this blue color dot-like structures, this is called as ribosomes. Okay, and this ribosomes will help in the protein synthesis. That is, they help in synthesis of proteins. Okay, and then next is intracytoplasmic inclusions. So these are inclusion bodies. So as you can see here, see, these are the inclusion bodies. All these oval shaped structures over here are inclusion bodies. And what is the function of these? These are the source of stored energy and present in some species of bacteria. Okay, so these cytoplasmic inclusions, they mainly store the energy. Okay, they are energy reserves. They store energy like glycogen, and so this glycogen or the energy can be utilized for the later cellular processes. Okay, and it's not present in all the bacteria, it's only present in some species of bacteria. And then the next one is mesosomes. Mesosomes are vesicular, multilaminated or convoluted structures found in the cytoplasm. They are centers of respiratory enzymes and prominent in gram-positive bacteria. There are two types of mesosomes, septal and lateral. So yes, as you can see here, so this part of the cell is called as mesosomes. Okay, so here the invaginations of the cytoplasmic membrane or plasma membrane is mesosomes. Okay, and so mesosomes are vesicular. Okay, they are vesicle like. They are multi-laminated, means they are made up of multiple layers, and they are convoluted, means they are coiled structures. Okay, and found in the cytoplasm. So they are also found in the cytoplasm, and they contain respiratory enzymes. Okay, respiratory enzymes means, so these are the enzymes which help in cellular respiration. That is nothing but breaking glucose to produce energy. Yeah, and they are mainly present in the gram-positive bacteria. And mesosomes can be of two types actually. One is septal and one is lateral. Okay, and as the name tells here, septal means septal mesosomes actually connect the plasma membrane to the nucleoid. Okay, so if the mesosome is present here, they connect this plasma membrane to the nucleoid. There will be connection with both the things. So because they play a role in cell division. Okay, and they also help in septum formation and DNA replication, and that's the reason they will have connection between the plasma membrane and the nucleoid. Whereas lateral mesosomes, they do not have any connections here. See here, as you can see, there is no connection with the nucleoid, and these are called as lateral. Okay, and mainly the respiratory enzymes will be present here. So these are the two types of mesosomes. Then next is nucleus. So this is a nuclear region. Okay, So bacterial nucleus has no nuclear membrane. So yes, so it doesn't have a nuclear membrane here. You can see that DNA is lying in the cytoplasm. Okay, so there is no membrane for this nucleus. Hence we call it as nucleoid. Okay, and the genomic DNA is double stranded in the form of circles. So yes, this is actually, this DNA is double stranded, and so it is in the form of circles. Okay, and the bacterial DNA maintains bacterial genetic characteristics. So yes, all the genetic characteristics is stored in the DNA. And then some bacteria may possess extra nuclear genetic material in the cytoplasm consisting of DNA named as plasmids. Okay, so that is apart from the genetic DNA over here, so some bacteria will also contain an extra nuclear genetic material. Okay, and this genetic material is called as plasmids. So as you can see here, so this is called as plasmids. So plasmids will not be present in all the bacteria. Some bacteria possess this extra nuclear genetic material which is called as plasmids. Okay, and the next, the outermost layer is capsule. So bacterial capsule or slime layer. It is the amorphous viscid bacterial secretion which surrounds some bacteria on the outermost layer. Okay, so here amorphous means there's no particular shape. Okay, and viscid means a glue like substance. Okay, so this glue like substance, no, they'll be present around the bacteria. As you can see, this is the outermost layer, and they may be present loose also, and they may be organized, and they may be sharp also. Okay, now based on that, when it remains as a loose secretion, unorganized secretion is slime layer. So you can see the difference in this picture over here in this. So the capsule, no, so they are not well organized. Okay, and it appears to be very loose. Okay, when it is like this, you call it as slime layer. And then in this picture you can see it is completely well organized, and it is sharp, and it is well defined also. Such structures are called as capsule. Okay, then okay, and some of the main functions of capsule is it protects the bacterium from phagocytosis. So means phagocytosis is a process of engulfing. So when a particular bacteria, no, when it enters a human body, our immune system will produce certain immune cells, so which can actually go, they can engulf and they can digest the bacterium. Okay, but when the bacterium have this capsule, no, it protects the bacterium. So it protects in such a way that the immune cell cannot engulf them and they cannot digest them very easily. Okay, so that is the function of capsule. It protects the bacterium. And then secondly, it acts protective covering against antibacterial substance, and it also protects against antibacterial substances. Antibacterial substances means the substances which kill or inhibit the growth of the bacteria. So it can also be medicines. Okay, when capsules are there, it's difficult to inhibit their growth or to kill, so hence they offer the protection. So these are some of the important functions of capsule. And then the next part is the flagella. Okay, as you can see, this is the flagella, and so different bacteria contains different types of flagella. So they are cytoplasmic appendages protruding through the cell wall. Okay, so as you can see, these are the appendages which come out from the cell wall. Okay, and so these are mainly responsible for the locomotion. That is, only with the help of flagella, a bacterium can move from one place to another place. They show motility because of this flagella. And they are composed of a protein called as flagellin. So yes, this flagella is made up of a protein called as flagellin. And there are four unique kinds of flagella. There are four kinds of flagella. First is monotrichous flagella. Okay, as the name tells here, mono means single. Okay, a solitary flagellum that lies at one end or the other. So these are called the polar flagellum. So as you can see in the picture here, so there is a single flagellum here. Mono means single, and there is a single flagellum at one end. Okay, it is called as monotrichous flagellum, and this flagella is called as polar flagellum. Okay, and then second is peritrichous flagella. A number of flagella are connected to the organism. They are in polar flagella as they are around the organism. Okay, so this is peritrichous. So as you can see here, entirely it is covered with flagella. Okay, and these are not called as polar because it's completely surrounded. Okay, and then lophotrichous flagella, a variety of flagella at one end of the microorganism, or on the contrary, they are described as polar flagellum. So yes, here this is amphitrichous and this is lophotrichous. Okay, in lophotrichous, as you can see here, they are present in one end. Okay, a bunch of flagella is present in one end. So hence they are also called as polar flagellum. When they are present in end, you call it as polar. Okay, and so this is lophotrichous. When they are present in only one end but a bunch is present, you call it as lophotrichous. And then next is amphitrichous. Solitary flagellum at both ends of the microorganism is known as polar flagellum. Okay, amphitrichous is again a single may be present or a bunch of the flagellum may be present on both the ends. Okay, and it's called as amphitrichous, and since they are present in end, they are called as polar flagellum. So next is main parts of flagella. So yes, as you can see in the picture here, so this is the structure of a single flagellum. So in the previous slide we studied that flagella can be single, it can be present in a group, or it can be present throughout the surface of the organism, right? So here in this picture you can see just the single flagellum. See, the flagellum will move like this, and these are some of the parts of the flagellum. Okay, and flagella mainly contains three main parts, that is basal body, hook and filament. Okay, so as you can see in the picture over here, so this part which is below, so this part is called as basal body. Okay, and this part which you can see here, this is the hook, and filament is a long hair like structure which you can see from outside. Okay, so this is how the flagella looks, and these are the main parts. So we'll study one by one. So first is basal body. So this part, the basal body is the anchor of the flagellum embedded in the cell's plasma membrane and cell wall. It functions as a motor that drives the rotation of the flagellum, allowing the organism to move. Yes, so it is the one which helps in anchorage. That means it is the one which is holding the flagella. Okay, and it is the one which is giving support to the flagella. So, and it is embedded where it's present and embedded in the cell's plasma membrane and the cell wall. Okay, and it is a one which allows the flagellum to move and make the bacterium also to move. And the basal body is composed of a series of rings that provide stability and support to the flagellum's movement. Yes, so as you can see here, there is one ring here, there is another ring here, there is another ring here, like this. There are series of rings, so in basal body, so which provides stability and it supports the movement of the flagella. Okay, so this is about basal body. And then next, this part is the hook. Hook is a curved structure that connects the basal body to the filament. It acts as a flexible joint, allowing the filament to rotate freely. This flexibility is essential for the effective propulsion of the organism through its environment. Yes. Okay, and this part is the one which connects the filament and the basal body. Right, so here is the filament and this is a basal body and it's present in between. So it is the one which connects the filament and the basal body. Okay, and it is actually a flexible joint. So hence it allows the filament to rotate very easily. Okay, and I can tell it is the one which acts as a driving force, and it also can push it forward. So So this flexibility is the one which is allowing the flagellum to move it easily. Okay, next part is the filament. Okay, filament is the longest part, so which we can see through the microscope. The filament is the longest part of the flagellum extending outwards from the hook. So yes, as I told, it's the longest part which we can see, and it is primarily made up of a protein called flagellin. So yes, so this is the filament is made up of a protein which is called as flagellin, so which forms a helical structure. The filament's rotation, driven by the basal body, propels the organism forward, enabling movement. So yes, as the filament starts moving, as it starts rotating, it allows the organism also to move forward. And then these three components work together to provide motility in various organisms, particularly in bacteria, where flagella play a vital role in their ability to navigate through different environments. So yes, so all these three work together, that is basal body, hook and filament, all work together and they bring about the movement in the flagella, so which in turn will help the bacterium to move. Okay, and flagella is one of the plays one of the vital role in movement, in motility of the bacteria. Okay, then next is Fimria. Found mainly in gram-negative organisms, Fimria or pili are hair-like filaments that extends from the cell membrane into the external environment. So yes, so apart from the flagella, so there is certain other hair-like appendages. So in the gram-negative organisms, which we call it as Fimria or pili. Okay, so when we compare this Fimria and pili is a little longer. Okay, when compared to Fimria. Fimria is a small hair-like structures which is surrounding the bacteria, and the main purpose of it is anchorage. They help in attaching the bacteria onto the surface. Okay, and then a, a pilus is composed of subunits, the protein pillin. It is made up of protein pillin. Okay, and the fimbriae are shorter, straighter, and more numerous than bacterial flagella. Yeah, it is very shorter and it will be straighter, and number will be more than the bacterial flagella. Okay, flagella will be very bigger when compared to fimbriae. Okay, and mainly, as I told, they are used in adherence to overcome the body's defense mechanism and cause the disease. So yes, they mainly help in anchorage and adherence. Okay, and then next is sex pili, which is also called as conjugation tube. So this pili is a little longer when compared to fimbriae, and the purpose of this is conjugation. Okay, that is, as you can see in the picture here, so the small hair like structure is over here is fimbriae, whereas here see this long one, so which is attaching to another cell, no, so this is actually called as pilus. Okay, it is specialized kind of pili that forms the attachment between the male and the female bacteria. Yeah, one is male and one is female here, and as in so one organism comes closer, the pili will grow in size, they will actually attach, and the transfer of DNA will happen here. Okay, see, during conjugation and acts as a conduct for the passage of the DNA. So this process is well characterized in gram-negative Bacillus Escherichia coli. Okay, this is one example where in Escherichia coli such thing will happen, process will happen. Then okay, next is endospores. Spores are highly resistant resting stage formed in unfavorable conditions, that is environmental conditions. As bacterial spores are formed within the parent cell, these are called endospores. So yes, the spores, these are nothing but spores which are formed during unfavorable environmental conditions. Like for example, when there is extreme temperatures, when there is drought, lack of nutrients, no, to survive, so they release the spores which is called as endospores. Why is it called as endo? Because it is formed inside within the parent cell, as you can see in the picture here, and that's the reason we call it as endospores. Okay, and then bacterial cells undergo spore formation in nutritionally deprived conditions, and this process is called sporulation. So yes, as I told, in the unfavorable condition or in the nutrient deprived condition, no, so this endospores will be formed, and the process of formation of this spores is called as sporulation. Okay, and these are highly resistant resting stages. So that means they will not be metabolically active. Okay, and so they will be highly resistant also. And and this endospores can also transfer, or they can come back into the active vegetative state when they get the suitable environmental conditions. Okay, and bacterial endospores can be visualized using a differential stain called as an endospore stain. Endospores can form in the terminal, subterminal and central regions of vegetative cells. These spores are metabolically inactive and highly resistant. So yes, so to visualize them, no, you need to do a special staining technique, that is a differential staining technique, which is called as endospore stain. And endospores, no, it can be in the terminal position, terminal in the sense in the end. As you can see here, this is the organism and the endospore is in the end. It can be in the terminal position, subterminal in the sense it's not completely towards the end but a little apart from the end. Okay, and then it can be in the central region also. So it can be in the terminal region, subterminal region and the central region. Then these spores are metabolically inactive and highly resistant. As I told, they are metabolically completely inactive and they will be highly resistant. Then, okay, next is the structure of endospore. Okay, so as you can see here, so this is the endospore and this is the spore mother cell or the vegetative cell. Okay, so and it has, endospore has different layers here, so we'll study them. Electron micrographs show that the structure of an endospore is very complicated. The exosporium is a thin fragile covering that is often around the spore. So yes, the outermost coat is the exosporium. They are very thin and they are fragile. Okay, then under the exosporium is a spore coat which is made up of several layers of protein and can be quite thick. It makes the spore resistant to chemicals because many toxin molecules can't enter through it, and it is also thought that the spore coat has enzymes that help the seeds to grow. So yes, under the exosporium is a spore coat. They are thick when compared to exosporium. Okay, and it is told that it is made up of several layers of proteins. Fine, and then the spore coat is the one which makes the endospore resistant because it does not allow the chemicals to enter in. Okay, and it is also told that it has certain enzymes which makes the endospores to grow. Fine, and then under the spore coat is a cortex which can take as much as half of the spore space. It is made up of peptidoglycan layer that is less cross-linked than that in the vegetative cells. Okay, now under spore coat, no, so this pink color area is called as cortex. Okay, and it is thicker. It is thick and it takes as much as half of the spore space here, and it is made up of peptidoglycan which has very less cross linkages. Okay, then the spore cell wall, also called as a core wall, is inside the cortex and surrounds the protoplast, which is also called the spore core. So yes, so now under this cortex, no, so there is core wall. Okay, so this core wall is the one which is surrounding the protoplasm. So this is a protoplast here. So protoplast is being surrounded by the cortex. So this blue color thing is protoplast and that is being surrounded by red color layer which is called as core wall. Okay, so we also call it a spore core. Then the core has normal cell parts like ribosomes and nucleoid, but it doesn't do anything metabolically. Now in this core, no, this is the center part which is called as a core, and in this core, no, there is ribosomes present, there is nucleoid, that is DNA present, but they are not metabolically active. Okay, so this is the structure of endospore.